Detection methods for microorganisms is being presented by Abhinav Datta, Prince Kumar Dubey, Abhinav Borua, Devashish Deori Bhorali, Hemanga Bijoy Baisya. Introduction. Quality of some food is also related to the number and the types of microorganisms present in the food. The detection and enumeration of pathogens in food and on the surface that comes into contact with the food are important components of any integrated program to ensure the safety of food throughout the food supply chain. Advances in technology have made the detection and identification faster, more sensitive, more specific, and more convenient. Importance. Important for safety point of view, as food may act as a vehicle for transmission of pathogenic microorganisms. Rapid detection methods are important particularly in food industry, as they are able to detect the presence of pathogens in raw and processed food immediately. Rapid methods are also sensitive enough to detect pathogens that present in low number in food. There are different detection methods for microorganisms. Types of methods are culture, microscopic and sampling method, chemical method, immunological method, molecular genetic method, and physical method. Culture, microscopic and sampling method. The examination of food for the presence, types and number of microorganisms and their product is basic to food microbiology. In spite of the importance of this, none of the method is common use permits the determination of exact number of microorganisms in food product. The four basic methods under this are as follows. Number 1. Standard plate count or aerobic plate count for viable cells or colony forming units. Number 2. The most probable numbers method as a statistical determination of the viable cell. Number 3. Direct microscopic count for both viable and non-viable cell. Number 4. Dye reduction technique to estimate number of viable cells that possesses reduction capacities.
And now we'll move on to the standard plate count or aerobic plate count for visible cells or colony forming unit. In this method, food samples are blended and homogenized in an appropriate diluent. This is then followed by serial dilution of the samples, after which plating is done in a suitable agar medium. And that is followed by incubation at an appropriate temperature for a given time. And finally, colonies are counted. And now we'll move on to the topic of plating, which can be categorized into two methodologies. Firstly, pour plate. In this method, appropriate dilution is added to the Petri plate, followed by the pouring of molten medium. The plate is then swirled so as to mix the agar and the diluent, followed by the solidification and incubation. Secondly comes the method of spread plate. Here, appropriate dilution is added onto the solidified medium in a Petri plate. The dilution is then spread on the surface of the plate using the spreader. Plate is then dried and incubated. The picture below exhibits both the pour plate method and the spread plate method. This image describes briefly the conventional SPC method. The first step would be to blend or homogenize portions of food samples, and then they are serially diluted in an appropriate diluent, which is then plated onto a single agar medium. And the fourth step would be to incubate them at an appropriate temperature for a given time. After that, all visible colonies are counted by the use of a cubic or an electronic counter. The SPC method is by far the most popular method for the determination of the number of viable cells or colony forming units in food products. This picture briefly describes both the spread plate method and the pour plate method. For the spread plate method, the first step would be to pipette the sample onto the surface of agar plate, and then sample is spread evenly over the surface of agar using sterile glass spreader. After that, incubation is performed, and finally, after which typical spread plate results can be clearly observed. Secondly comes the pour plate method. Here, sample is pipetted into the sterile plate, and then the sample medium is added and mixed well with the inoculum. After that, incubation is similarly performed, and finally, after which typical pour plate results can be clearly observed. We will now move on to the colony counts. Here, number of colonies are counted. Plates that have a range of 30 to 300 colonies are used for calculating the results. But the plates that have more than 300 colonies are considered too numerous to count, following which calculations are performed. The formula for which is colony forming units divided by the volume of dilution plated. Here, colony forming unit stands for the number of colonies multiplied by the dilution factor. And now comes the spiral plater. The spiral plater is a mechanical device that distributes the liquid inoculums on the surface of a rotating plate that contains a suitable poured and hardened agar medium. The dispensing arm moves from the near center of the plate towards the outside, depositing the sample in an Archimedes spiral. The attached special syringes dispense a continuously decreasing volume of a sample so that a concentration range of up to 10,000 to 1 is effected on a single plate. Following incubation at an appropriate temperature, colony development reveals a higher density of deposited cells near the center of the plate, which progressively veer toward the edge, which is very clearly described by the picture below.
The advantages of the spiral platter are: here less agar is used, fewer plates, dilution banks and pipettes are also required. 3 to 4 times more samples per hour can be examined. There are some disadvantages in this method too. Food particles may cause the blocking in the dispensing stylus. Membranes with a pore size that will retain bacteria but allow water or diluents to pass are used. Following the collection of bacteria upon filtering a given volume, the membrane is placed on an agar plate or an absorbent pad saturated with the culture medium of choice and incubated appropriately. Following growth, the colonies are enumerated. Cellulose filters were among the earliest used. However, polycarbonate nucleopore filters offer the advantage of retaining all bacteria on the filter. Now the most probable number method, or the MPN method. The most probable number, or the MPN, is a statistical method used to estimate the viable numbers of bacteria in a sample. It is often used in estimating bacterial cells in water and food. However, this method is only capable of enumerating viable organisms. Some of the advantages include: specific group of organisms can be determined by use of appropriate selective and differential media. Its method of choice for determining fecal coliform densities. Some of the disadvantages are: large volume of glassware is required in this method. The lack of opportunity to see the morphology of the colonized organism. Lack of precision. Now moving on to the next method called the direct microscopic count, or the DMC method. This method is very simple and rapid method for the initial morphological identification and count of bacteria and molds. Morphologically, bacteria are classified into different groups. Mainly, there are 3 main types of different microscopic count methods. Number 1, breed count. Number 2, slide method using INT. And the third one is Howard mold counts. The advantages of DMC method are: rapid, simple and easy method requiring minimum equipment. Number 2, morphology of the bacteria can be observed as they counted. Here, very dense suspension can be counted if they are diluted appropriately. Next, we have the dye reduction technique. Dye reduction test is a common technique used to detect the microorganisms from food. Here, 2 dyes are commonly employed in this procedure to estimate the number of viable organisms in suitable products: methylene blue and residuine. To conduct a dye reduction test, the supernatant of food is prepared and added to standard solutions of either dye for reduction from blue to white for methylene blue and from slate pink to white for residuine. Two dyes are commonly employed in this procedure to estimate the number of viable organisms in suitable products. To conduct a dye reduction test properly, prepared supernatant of food are added to standard solutions of dye for reduction, from blue to white for methylene blue and for slate pink to blue for white for residuing. The time for dye reduction to occur is inversely proportional to the number of organisms in the sample. Now the chemical methods. The chemical methods used primarily to detect, enumerate or identify foodborne organisms of their products are: number 1, thermostable nucleus; number 2, Limulus amoebocyte lysate assay for gram-negative bacteria. Then we have the ATP assay. Then we have radiometry, and lastly fluorogenic or chromogenic substrate. Now the number 1 chemical method is the thermostable nucleus. The presence of S. aureus in significant numbers in a food can be determined by examining the food for the presence of thermostable nucleus. This is possible because of the high correlation between the production coagulase and the most stable nucleus by S. aureus, especially enterotoxin producers. It has been found to be as good as coagulase in testing for enterotoxigenic strains. Aureus, especially enterotoxin producers. It has been found to be as good as coagulase in testing for enterotoxigenic strains.
Litmulus lysate for endotoxins. The Limulus amoebocyte lysate test employs a lysate protein obtained from the blood cells of the horseshoe crab. The lysate protein is the most sensitive substance known for endotoxins, and the presence of endotoxins causes gel formation of the lysate material. LAL test is a good rapid indicator of the total numbers of gram-negative bacteria. The method has been applied successfully to monitor milk and milk products, microbial quality of raw fish, and cooked turkey rolls. Adenosine triphosphate measurement. Simplest way to measure ATP is by the use of Firefly luciferin luciferase system. In the presence of ATP, luciferase emits light, which is measured with a luminometer. The amount of light produced by the Firefly luciferase is directly proportional to the ATP added. It has been adapted for the determination of microbial load on chicken carcasses as well as pork and beef. It is widely used as a rapid and on the spot method for monitoring food handling surfaces by swabbing designated areas and reading the relative light unit from a luminometer. The diagram shows the process of adenosine triphosphate measurement. Next, we move on to the topic radiometry. The radiometric detection of microorganisms is based on the incorporation of a C-labeled metabolite in a growth medium, so that when the organisms utilize this metabolite, CO2 is released and measured by the use of a radioactivity counter. For organisms that utilize glucose, C-glucose is usually employed. For those that cannot utilize this compound, others such as C-formate or C-glutamate are used. The time required to detect the labeled CO2 is inversely proportional to the number of organisms in a product. Fluorogenic and chromogenic substrates. Some of the fluorogenic and chromogenic substrates employed in the culture media in food microbiology are 4-MUG, 4-MUGAL, MUP, ONPG, LAPN, 5-XGICA, 5-XGAL, IBDG. Next, we move on to the topic immunological methods. Immunological methods involve the interaction of a microbial antigen with an antibody that is produced by the host immune system. Testing for microbial antigen or the production of antibodies is often easier than test for the microbe itself. Lab kits based on this technique is available for the identification of many microorganisms. Serotyping. Serotyping is most widely applied to gram-negative bacteria entering the bacterial pathogens such as Salmonella and Escherichia. Among gram-positives, serotyping is important for the genus Listeria. The gist of a typical serotyping scheme is the use of specific antibodies or antiserum to identify homologous antigens. This type of scheme results in the recognition of three antigenic sites. First one is somatic antigen, second is capsular antigen, and third flagellar antigen. Different techniques under immunological methods: fluorescent antibody, enrichment serology, Salmonella 1 to 2 test, radioimmunoassay, ELISA, gel diffusion, immunomagnetic separation, and hemagglutination. The chart shows the immunological test and some of their uses.
Molecular genetic methods. The problems associated with culture-based methods for detection and identification of microorganisms in clinical and environmental samples have motivated the development of alternative methods which do not require cultivation of the target organisms. Molecular genetic methods which specially target microbial nucleic acids which have become an important tool for the identification of both culture and uncultured microorganisms. Molecular genetic methods have become a powerful set of tools used by many non-culturable states as well as organisms which have yet to be brought into culture. Technological innovation in the detection and identification of the microorganisms using molecular techniques such as polymerase chain reaction have ushered in a new era with respect to diagnostic microbiology. Fingerprinting methods. Currently, the most widely accepted of these techniques involves restriction endonuclease cleavage of the whole genome with rare cutting enzymes, followed by separation of the resulting large DNA fragments by pulse fluid gel electrophoresis. The number and size distribution of these fragments is commonly referred to as fingerprint and is normally characteristic for different strains. There are different types of fingerprinting. It includes phase typing, amplified fragment length polymorphism, multicolors enzyme electrophoresis, restriction enzyme analysis, restriction fragment length polymorphism, ribotyping, microarrays. So physical methods, biosensors. A device, it contains the biological sensing element connected to a transducer, enzyme, substrate or antigen antibody reaction may be the components of biosensor. Biosensor that have been demonstrated to be the value of for food-borne microorganisms. So it has three subcategories. Firstly, acoustical biosensors. With the quartz coated with the antibody, the targeted analyte is the homologous antigen which, when it binds to the antibody, changes the mass into a resulting decrease in frequency. Secondly, fiber optics. A fiber optics biosensor uses electronic or optical transduction to monitor a biological reaction and reports it to an optical signal. Thirdly, impedance. When microorganisms grow in cultured media, they metabolize substrate of low conductivity into products of higher conductivity and therefore decreases the impedance of the media. Microcalorimetry. This is the study of small heat changes, measurement of the enthalpy change involved in the breakdown of growth substrate. The heat production of this measurement is closely related to the cell's catabolic activities. Secondly, flow cytometry. It is the science of measuring components and the properties of individual cells in liquid suspension. Thirdly, biosys instrument. The biosys 32 instrument makes automatic and computer analyze change into the color of reaction vials as organisms grow in specified culture media that contain substrate that changes color proportion to increase the number of cells. It usually used to detect salmonella and listeria, etc. So, flow cytometry. Flow cytometry is a technique that is used to detect and measure physical and chemical characteristics of population of cells or particles. In this process, a sample containing cells or particles is suspended into a fluid and injected into the flow cytometer instrument. So, conclusion: assurance of food safety, particularly regarding microbial contaminants, is still a very complex task. They require some additional preparation that may prove to be a little too expensive for mainstream use. Numerous intricate studies and new fungal methods and procedures are being used that could have a serious chance of proving to be excellent in detection microbials and bacterial contaminants. The references from where we collected the information is given here. Thanks for watching the video. Don't forget to like, share and subscribe.